Hi there, Queen Nerdling here, and today we're going to be talking about proteins. Let's get started. So first of all, the monomers of proteins are amino acids, and the polymers are the polypeptide chain or the protein itself. Now the type of bonds that link those amino acids together are called peptide bonds. And together, if we're pretending that each one of these guys is an amino acid, we call this a polypeptide chain. So as far as the structure of amino acids, we have a couple of different components. We have this centralized carbon right here. We have an amine group, which is composed of a nitrogen and two hydrogens, or NH2, if you remember your functional groups from the elements of life. We also have a carboxyl group, or that COOH, and we have a variable R group. Now that variable R group is what differentiates one amino acid from the next. And there are 20 different varieties of amino acids that compose all of the proteins in living organisms. So in most biological systems, like I said, we have 20 different types of amino acids. And don't worry, you do not need to memorize these for any tests unless you're a biochemistry major in college. So proteins have many different functions, and they also have a ton of different structures. And if you remember, the structure dictates the function of an organism or an organelle or a molecule. So as far as functions, proteins can serve as enzymes in the form of pepsin or DNA polymerase. They can form structures such as keratin that's composing my hair, collagen that helps keep us all plumped up. They can serve for carriers and transport. They can serve in cellular communication as both the signals themselves as well as the receiving protein. They can serve for defense. They can also serve for movement in our actin and our myosin muscles. So basically like contracting my muscles right here. They serve for contraction and they compose those muscle fibers. They also serve for storage, such as in beans, seeds, the ovalbumin in eggs. So let's take a little bit more of an in-depth look about all of those functions that the different protein structures have. So obviously enzymes are very important for life. Enzymes are responsible for basically all of those reactions that are going on in our body. So enzymes in our metabolism work to break larger polymers down into substances that we can utilize in our body. They're also responsible for building or for putting different molecules together for storage purposes. Now, enzymes are what we call catalysts. They catalyze or speed up reactions by lowering the activation energy. Enzymes are specific for the reaction in which they catalyze, and they can either be classified as catabolic, by scratching those bonds apart through hydrolysis, or anabolic, meaning ana adds, through the process of dehydration synthesis. So proteins also serve storage purposes. So for example, we have casein, which is just a protein in milk. It's a major source of amino acids for baby mammals, including me at one point in time and you at one point in time. Plants also have storage proteins in their seeds. We have ovalbumin, which is the storage protein in egg whites, and it's used as an amino acid source for the developing embryos, as well as for me when I eat them in the morning. Proteins also serve hormonal functions, so we do have hormonal proteins. It helps to coordinate organisms' activities. So for example, insulin is secreted by the pancreas. This causes other tissues to take up glucose, which helps to lower our blood glucose level. And if you have an abnormality with your receiving proteins, then you might have high blood glucose levels and you might have to go on insulin. And we call that diabetes. There are two different types, type 1 and type 2 diabetes, and we'll be discussing those in a different lecture. So proteins can also serve for defense against invaders, such as viruses and bacteria. So they form these structures that look kind of like Ys, and they're called antibodies. Now, antibodies have an antigen binding site. Now, antigens are bad guys. They are the viruses, such as this guy that's wreaking havoc everywhere around the world right now. And they bind with those antigens, and they either destroy or inactivate them. So another function that proteins have are transport. So they can transport substances into and out of the cell. Sometimes they form channels, such as aquaporins, that can allow water through a cell membrane. They also can transport different substances in our blood. So for example, hemoglobin is an iron-containing protein in vertebrate blood. I'm a vertebrate. Anything that has a backbone is a vertebrate. And it transports oxygen from the lungs to other parts of the body that are going to need it. Especially some of our muscles that are working really, really hard. We need more oxygen in those muscles in order to have better energy levels and better function. They also serve in cell signaling purposes. So they can compose receptor proteins, like these guys down here. They're responsible for receiving the signal, or they can be a signaling molecule themselves. And for example, this is a nerve cell. So this is going to be an axon. This little space in between is called a synapse, meaning the space between two neurons. Now we have these little signaling molecules, which can be proteins, and then the receiving protein on the other axon or the other nerve cell that are basically like these little catchers that receive that signal, and then they'll transmit it down to the next nerve cell until we eventually will get a response. They serve for structural support. So example, like I said earlier, keratin, which composes our hair, fingernails, horns of different animals, insects, appendages. Spiders use it for their silk filaments, cocoons, and then we also use it for collagen and elastin proteins. And those provide those fibrous frameworks in animals' connective tissue. And so as we age, we actually start to lose some of our collagen fibers. We start to get wrinkles. So that's kind of what helps us keep plumped up. And I'm sure that most of you have heard about people getting collagen injections in their lips, or a bunch of different types of facial products that are supposed to stimulate collagen production, or laser treatments that stimulate collagen production, because we're constantly trying to rebuild that structure of our skin so we can remain nice and youthful like you. So in proteins, there's a very specific order of the amino acids that dictates the structure. And remember that that structure of the protein is going to determine its function, meaning what does it do, what does it become, and how does it help or hurt us. So amino acids have directionality, meaning they have to be in a specific sequence in order to form a very fully functional protein. Now there are 4 levels of protein structure. We have primary, secondary, tertiary, and quaternary. And each level is determined due to different interactions among amino acids. So the primary structure is simply the amino acids all linked together through peptide bonds. Just the sequence of those amino acids, so one amino acid coming to the next through a peptide bond, and it forms a polypeptide chain, bunch of amino acids linked together through peptide bonds. Now we also have a secondary structure. So once we've had that primary structure, we get a secondary structure. And there are 2 different types of secondary protein structures. We have alpha helices, very similar shape to DNA or our nucleic acids. And then we have these beta pleated sheets that are kind of like this up, down, beta pleated sheet. Now in the secondary structures, we have interactions between hydrogen bonds. So hydrogen bonds would be linking the little rings in the alpha helices together, and it would be linking those beta pleated sheets together as well. Now comes the messy part. The tertiary structure is basically a bunch of alpha helices and beta pleated sheets that get twisted and turned together, and they start forming stronger ionic bonds as well as stronger covalent bonds. Now we have ionic bonds that are getting put together with different types of salts. We have hydrophobic and hydrophilic interactions. And we also have very strong covalent bonds that are called disulfide bridges, meaning two sulfurs, disulfide, so disulfide bridges. Now, not every tertiary structure is going to look this crazy. So for example, the collagen fibers, we're just going to have a bunch of alpha helices together. We could also have a bunch of beta-pleated sheets put together. But many proteins have a combination of beta-pleated sheets and our alpha helices that get turned into this squiggly mess that you see on the screen in front of you. Now we can also have a quaternary structure. That is when two or more tertiary structures come together to form a larger functioning protein. One of the best examples is hemoglobin, which we talked about earlier. Now this is an iron-rich compound that helps to transport oxygen from the lungs to other areas in the body that need it. You knew better than know you wouldn't get out of one of my lectures without seeing me demo things with tangle beads. So I wanted to demonstrate the 4 different structural levels or the 4 different conformations of a protein using these guys. Now, first of all, we have that primary structure, and that is just the sequence of amino acids. And if we're working with this, we are going to pretend that each one of these different little tangle beads is an amino acid, and those are held together through peptide bonds. Now, many amino acids linked together through peptide bonds are called a polypeptide chain. And this is also the primary structure or primary conformation, meaning shape, of a protein. So primary structure. Now, the secondary structure, we can either have alpha helices, so I can kind of twist this around my finger. We get an alpha helices, very similar in structure to a DNA strand. Now, we will get hydrogen bonds that form between the rings of this structure, of the secondary structure. So this is one of 2 of the secondary structures that you can have in a protein. So we have alpha helices, and the second type is called a beta-pleated sheet. So the beta-pleated sheet is basically just kind of this little up, down, squiggly. And again, we're going to connect them through hydrogen bonds. We're not going to have any interactions between the R groups quite yet. Now, the tertiary structure, or that 3rd level of a protein, is going to be when we start getting those interactions between the R groups. We start having those stronger covalent bonds called disulfide bridges that occur, and we also start getting ionic bonds. So we might have a combination of a alpha helices and then a beta-pleated sheet. And they start to twist in on each other. And so again, in this tertiary structure, we now have interactions between those R groups. So we start to form ionic bonds, like salt-type bonds, hydrophilic and hydrophobic interactions. And we also start to get those really strong covalent bonds. And those are called disulfide bridges, meaning 2 sulfides connected together. Now, if we go on to the quaternary structure, we would have 2 or more of these tertiary structures that come together. And so the example that you just saw was hemoglobin. So we'd have 4 of these tertiary structures that come together to form a hemoglobin molecule. But most of the time, the tertiary structure is that fully functional protein. So again, we have primary, secondary, tertiary, and sometimes quaternary. And sometimes we might expose those enzymes or proteins, whatever we're working with as far as the protein's concerned, to extreme conditions such as high heat or pH. And you know what happens? A lot of those bonds get broken, and it will go back into its primary conformation of just that strand of amino acids linked together through peptide bonds. And we call that denaturation. That's whenever we're breaking all of those bonds, goes back to its primary conformation. So hopefully that little visual demo was somewhat helpful for you. And so this would be the example of that quaternary structure I just got through demoing with my tangle beads. If more than one of those tertiary structures came together. In fact, for hemoglobin, it would be 4 of these little squiggly guys. So what happens when there is a change in the structure of amino acids? Well, it's obviously going to impact or change the function. So one of the common examples of a change in the structure affecting the function of a protein is sickle cell disease. There is a very small change in the primary structure of that protein, meaning the primary structure of this. So it is caused by one single point mutation, meaning one teeny little tiny amino acid causes a huge change in the structure of that protein. And this can result in diseases. This example is sickle cell disease. It's an inherited blood disorder that results from just one single amino acid substitution in that protein hemoglobin. So we have one nucleotide base that gets subbed in. It causes the chain of amino acids to differ. So instead of having one amino acid, we have a different one. That changes the structure of the protein. When the structure of the protein is changed, it changes whatever molecule that it's going to be building. In this case, it changes our blood cells from pretty nice little discoid shapes to this sickle shape that can get stuck in our blood vessels. It can lead to clotting disorders. It doesn't transport oxygen as well, so people that have sickle cell disease tend to have a lot of issues with their lungs and breathing. And so we're going from glutamine to valine. So instead of it being glutamine, we now have valine, and that results in that structural change. And again, we're having a normal hemoglobin molecule. So once that happens, we have our normal hemoglobin. It results in this nice discoid red blood cell shape. Now, whenever we get that substitution mutation that occurs, we are now having valine substituted for glutamine, and it causes a conformational change, meaning a change in the shape or the structure of that protein. When that protein structure changes, it's going to change the function. And again, we go from a nice discoid shape now to that sickle shape. So like I showed you with the tangle beads, sometimes proteins undergo extreme conditions. So if we add extreme heat or acidity or salt levels, it causes what we call denaturation. So again, we go from nice little squiggly right here, denaturation occurs, and it goes back into that primary conformation. It will obviously disrupt the function of the protein. Now, many times whenever this happens, it can cause the protein to basically just go back into that primary conformation. And if it goes back into a favorable or ideal conditions, then it can renature, meaning go back into a functional protein. Now, sometimes we can actually break up those polypeptide chains, and in that case, that protein would be non-functional permanently. So I hope this was helpful. So just as far as a review and kind of like the take-home points that you should have from this, remember that proteins are composed of carbon, oxygen, hydrogen, nitrogen, and sulfur. They have those R groups, and that's what makes one amino acid different from the next. And we have 20 of them. The monomers of proteins are amino acids. The polymers are those fully functional proteins. It could be an enzyme, it could be a structural protein, it could be a cell signaling receiving protein, a cell signal itself, and then proteins function for building blocks, for cell-to-cell communication, for storage, for catalyzing different types of reactions. Basically, they are involved in all walks of what life is like inside of our bodies, in plant cells, animal cells, everything. Protein is responsible for everything, and that goes into the central dogma as well. So I hope that was helpful. If you enjoyed this video and you'd like more weird, quirky videos from the Queen Nerdling, you can go to www.nerdlingscience.com and find more videos. This is the Queen Nerdling signing off. I hope you have a great day.